Did you know that inside every single one of your cells, there's a history book? It's written in the language of molecules, and it tells these epic stories of survival and innovation that stretch back millions, even hundreds of millions of years. Today, we're going to read a few chapters from that book.
You know, it all comes down to these tiny, incredibly intricate machines called receptors. They're basically the senses of the cell, the gatekeepers that translate everything from the outside world into the language of biology. But here's the thing. These machines weren't designed on a drawing board. Nope, they were sculpted by evolution. Okay, let's really dive into this.
So here's the plan. We're going to take 3 distinct journeys deep into our evolutionary past. We'll uncover the origin stories for the receptors that let us see, smell, and even feel pain relief. This is our roadmap for today's explainer.
All right, let's start with the big picture. Receptors are just proteins, sitting on or inside our cells. And when a signal shows up, could be a photon of light, a hormone molecule, or even the active ingredient in a medicine, the receptor is what catches it and tells the cell, okay, here's what we do next. But here is the absolutely crucial point. These things are not static parts off an assembly line. Each one is like a living fossil, the end result of this long, messy, but brilliant process of genetic copying, pasting, and editing. Their history is literally our history, written right there in our DNA. Our first journey is going to take us all the way back to the dawn of sight. The amazing ability to see is powered by a whole family of receptors called opsins. These are the molecules that literally catch light. So how in the world did we get them?
Well, it all kicked off with one single ancestral gene. That gene got copied, creating duplicates, but the real game changer came when the entire genome of our very early vertebrate ancestors was duplicated not once but twice. Think about it like accidentally photocopying your entire library of instruction manuals. Suddenly, evolution had all these spare copies to just tinker with. This meant one copy could keep doing its original job, while the new one was free to evolve new functions. And that's how we got rhodopsin for night vision and a whole suite of other options for color vision.
And look at this. This is what all that tinkering produced. This is rhodopsin. It's a classic example of what's called a G-protein coupled receptor, or GPCR. You can see those 7 helices that kind of snake their way through the cell membrane. And tucked right inside is a molecule called retinol. It acts like a tiny antenna. The moment a photon of light hits it, that retinol snaps into a new shape, which forces the whole protein to change shape and send a signal. It is a breathtakingly elegant piece of molecular machinery. So this whole evolutionary process created 2 totally distinct systems in our eyes. We've got our rods, which use that super-sensitive rhodopsin, perfect for seeing in dim light. And then we have our cones, which use a bunch of different opsins, each tuned to different wavelengths of light, giving us our rich experience of color. 2 different tools for 2 different jobs, and all of it born from a single ancestral gene. Just incredible. All right, for our second journey, we're diving deep inside the cell's command center, the nucleus. We're looking at a family of receptors that act like master switches. These proteins read the body's internal mail, signals like hormones. And then they go and directly turn genes on or off, controlling pretty much our entire physiology. Now, the evolutionary story here is a little different. It's not just about copying things over and over. It's about a wild concept called domain swapping. The easiest way to think about it is like this. These proteins are built from molecular Lego bricks. One brick, or domain, is responsible for grabbing onto DNA. Another, different domain, is responsible for recognizing the hormone signal.
So evolution, acting like a master tinkerer, literally cut and pasted these domains between different ancestral genes. It could, for instance, take the DNA binding part from receptor A and snap it onto the signal binding part from receptor B, and poof, just like that, a brand new receptor is born with a totally new function. This is a principle nature figured out long before human engineers ever did: modular design. By mixing and matching parts you already know work, you can create new functions with incredible speed and efficiency.
And the result of this long history of duplication and domain swapping is the rich diversity of receptors we see today. Phylogenetic analysis clearly groups these master switches into 3 major subfamilies. You've got the ones that respond to thyroid hormones, the ones that respond to steroids like estrogen and testosterone, and then this really fascinating group called orphan receptors. We know they exist, but we're still figuring out what signals they're listening for. Okay, our final journey is about a sensory explosion. This is the story of how evolution rapidly built a massive library of receptors, giving our ancestors a rich sense of smell and, get this, new ways to regulate fundamental things like pain and reward.
So the key mechanism this time is something called birth and death evolution. It's not about subtle tweaks. We're talking explosive growth. A gene gets duplicated, then it gets duplicated again, and again, and again, rapidly creating this huge family of related genes. Now, some of these new copies gain new functions, while others pick up mutations and become inactive, little fossils in our genome, like silent echoes of past evolutionary experiments.
And we know this process is ancient. I mean, really ancient. Scientists have found over 30 different smell receptor genes in the lancelet. That's a tiny fish-like creature that's one of our most distant evolutionary cousins. Since it branched off our family tree over 500 million years ago, that tells us the genetic toolkit for a complex sense of smell was already well under construction way back then. Now, this exact same story of duplication and diversification gave us another critically important receptor family, the opioid receptors. An ancestral gene was duplicated during those same whole genome events we saw with vision, creating the 4 members we have today. And each one is tuned for a slightly different job, from the mu receptor's powerful role in pain relief and reward to the kappa receptor's involvement in stress. This, right here, is why different opioid drugs have such different effects. They're targeting different members of this ancient family. So we've taken 3 pretty different journeys, right? We looked at vision, hormonal control, and sensation. But when you pull back and look at the big picture, you can see they were all built using the same fundamental toolkit.
Let's just recap evolution's core creative processes. First up, gene duplication. This creates the raw material, the spare parts. Second, mutation and natural selection. This is what slowly sculpts those parts into new shapes and functions. And third, in some cases, you have domain swapping, which acts like this powerful shortcut, rapidly combining existing solutions to create totally novel innovations. These aren't just theories, you guys. These are the mechanisms written into the history of our own DNA.
So why does tracing all this deep history actually matter? I mean, it's fascinating for sure, but it's not just some academic exercise. Understanding where these molecular machines came from is absolutely critical to understanding how they work, and just as importantly, how they fail in disease. You see, this knowledge is at the very heart of modern medicine. When we design a new drug, what we're really doing is targeting a receptor that is part of an ancient family. Knowing that family tree, knowing which members are most closely related, helps us predict side effects. It lets us design more specific drugs that hit their intended target without messing with all of its evolutionary cousins. It basically transforms drug design from a shot in the dark into a predictive science. And make no mistake, this story is far from over. As this quote from just last year highlights, even for rhodopsin, one of the most studied receptors in all of biology, there are still deep mysteries to solve. We have this amazingly detailed picture of all the parts, but understanding how they all integrate together into a complete, living system, well, that's the next great frontier.
Which really leaves us with a final thought. We've just read 3 chapters from the history book inside of us. But there are thousands more pages, thousands of other gene families with their own incredible sagas of innovation and adaptation. What other secrets are just waiting in our DNA for us to decipher?